Good evening. Welcome to the second of this summer series of Wednesdays at the Genome. I'm Stan Fields, a faculty member in the Department of Genome Sciences. This is the 11th year that the department has organized a series of public seminars to help the wider community understand some of the research that's going on in genetics and genomics, and to gain some appreciation of the work that's going on in our department here in this building. We're delighted that you're joining us this evening, and we hope you'll stay after the talk for the coffee and cookies, and we can engage in some additional conversation. Our speaker tonight is Dr. Jay Shendure, Professor of Genome Sciences. Jay did his undergraduate work at Princeton, and then he did an MD PhD at Harvard University, where he worked with George Church, a distinguished leader in genetics and genomics. George happens to be in the back leaning against the pole, and he's visiting us coincidentally at this time. At Harvard, Jay was a key part of work in George's lab for some of the earliest efforts to make DNA sequencing approaches much more high throughput than they had ever been. And this has changed the way we think about genetics today, particularly true for cancer research. Jay came to the University of Washington in 2007 and has become a leader in this field, identifying gene mutations associated with disease and showing that fetal DNA can be sequenced using non-invasive approaches. He's won several major awards already, including innovation awards of 35 under 35 and 40 under 40. I don't know how long you can keep doing this, maybe 100 under 100 or whatever. Tonight he will be speaking on using CRISPR to learn how our body builds itself. Welcome, Jay.
Thanks. Thank you all for coming out tonight. It's kind of an odd kind of pressure here. I put these slides together in part with my kids in mind, who are here in the front row. And then my PhD advisor showed up, so I've got two audiences to serve here. So I'll do the best I can. I also feel like I'm defending again, which is an odd feeling. But, you can't fail me now. So okay, so I'm just going to start with two upfront acknowledgments. The first is Ed Young, who's a Atlantic writer who wrote about some of our work when the paper came out, and just I'm mostly crediting him with the title here, which was the title of his article. And the second person I'd like to credit is Aaron McKenna, who's Dr. Aaron McKenna, who just got his PhD last week. And so one of the dirty secrets of professors is that we get half our slides from our students. And so the nice coincidence of his thesis on this topic and this talk helped me a lot, and a lot of the slides that I'll show are borrowed from him. Okay, so I'm going to talk about this topic I'm going to break it into three parts. So the first part is a crash course in cell lineage, and I'll explain what I mean by cell lineage in a few slides. The second part is a crash course in CRISPR editing. I just realized I told Charles that I don't have sound, and I do. Is Charles still here? Yes, you're right there. Okay, sorry. And then the last part, I'll talk about the experiment that we actually did to try and put these two things together and use CRISPR genome editing to learn something about cell lineage. Okay, so what is cell lineage? So this is a very simple concept. So each of us, everyone in this room, started as a single cell, right? At the top of this tree, as shown here. That cell then divided to give rise to two cells, so one generation. Then another generation, you're at 4 cells. Another generation, you're at 8 cells, right? So it's kind of like a family tree of cells, right? This is asexual reproduction, so each cell is dividing into 2. Only has one parent. Each cell only has one parent, but has a grandparent and so on and so forth. This obviously keeps going. So after 4 generations, you're at 16 cells, and then 5 generations, 32 cells, but it starts to rise very quickly. And so once you're out to just 45 generations, you can have as many as 40 trillion cells, right? Which is approximately the number of cells in an adult human, right? Okay, and so when we talk about a cell lineage, we're talking about this tree, the set of relationships between cells with one another and their ancestors. So very analogous to a family tree, except the people are cells, and you just have this splitting at each generation. Okay, so it's obviously more complicated than just this. So that first cell is called a zygote, a fertilized egg. So I have a deal with my wife where she explains everything on this side to the kids, and I explain everything on this side. So I'm getting mine out of the way tonight, and she'll be. So there are fancy names, of course, for different parts of this. So once you start dividing up this little ball of cells, it's called a morula, and then it starts to not just be a amorphous blob here, but to actually have some structure and be called a blastocyst, and then it keeps dividing. And so when you get to this mass of 40 trillion cells that is an adult, every cell in an adult or a child has a history. So for example, if I was to look at a cell in the stomach, it had some set of ancestors that can be traced all the way back to that first cell. If I was to look at a cell in the brain, it has a different set of ancestors, but eventually it and the stomach cell have the same ancestor when you get way back here, and obviously the first cell is everyone's ancestor, right? And I look at, let's say, an eye cell, and maybe it also has a different history, but its history is very related to the history of the brain cell, right? And so when we're trying to learn about cell lineage, we're trying to learn about this set of relationships between different kinds of cells in the adults with one another as you go backwards in development, right? And so the crazy thing is that so this is true of every human here, right? And every human on the planet. But it's also true of every other animal and plant on this earth, right? So everything, every animal and plant all started from a single fertilized zygote, right? And so plants, flies, frogs, Barack Obama, jellyfish. I try to show myself pictures of Barack Obama frequently these days. So he's in the talk a lot. So meerkats, zebrafish, all go back to a single cell, right? A single fertilized egg. We all started out this way, and somehow remarkably, we gave rise to all these different things. So not just the different people you see in this room, but also all different kinds of life. And so the other thing I'd like to just note is that you look at the diversity of life, and it all looks cool and amazing, but a lot of what's really amazing about living things are things that we can't see, right? So here's an example. So I don't know if that image is coming out well there, but so someone managed to label the circulatory system or the blood vessels of a parrot, and so you can see all those blood vessels, right? That's the eye of a wasp. I could have had an entire talk just composed of cool images. This is the part of a mouse's brain that's involved in memory formation, showing connections between different neurons. And so you look at a fly or a mouse or a parrot, and you don't necessarily see all this really magical complexity that's inside every living thing, and all of the relationships between cells that kind of enable this all to work, right? Okay, so it's super complicated, I guess, is the main take home here. So how can we break it down into something that's a little more we can get our head around? Okay, so and also I guess the related question is why? If we go back to this slide, why does this one fertilized egg go on to become a fish, and another fertilized egg go on to become a jellyfish? What's the difference that makes it go this way instead of that way? So the difference is the genome, right? And so the genome is basically consists of this long string of A's, G's. You can turn the light down, actually. It'd be easier to see the slides. But can people see well, or better off or on? Off. Okay. long string of A's, G's, C's, and T's, right? So the human genome is about 3 billion letters in length. The genome of a worm or a fly is about 100 million letters in length, and the letters, the order of these letters is effectively a set of instructions for how to build a particular kind of thing, right? So a good analogy for this is just a recipe book, where you can have a recipe book with written letters on the page, and one recipe might tell you how to make cookies, a different recipe might tell you how to make something else. And so the language we use in genetics, so the equivalent of a recipe is a gene. And instead of telling you how to make a cookie, it's telling you how to make a protein, right? And that protein is kind of like a small molecular machine that can go and do something, like making part of a cell wall or something else. And so in total, the human genome and the fly genome and the worm genome each have about 20,000 recipes, right? So you can think of it as a book of about 20,000 recipes to make different kinds of proteins that are then used to build that living thing. So obviously, you need more than just a set of recipes. You need someone who can read the recipes, right? And so you also need a chef, right? And so in every cell in your body, there is both the genome, the recipe book, and there is a set of molecules that kind of act as the chef, so to speak, knowing how to read this instruction manual and build proteins, and actually build new copies of the chef, too, so you can propagate the chef. Okay. And and this is really the amazing thing, is that this whole genome is copied into every new cell, right? So as we go from 1 cell to 2 cells to 4 cells, the genome, the book, gets copied. The chef gets copied too, right? Makes a copy of himself, and so on and so forth. So you can imagine if this process was allowed to continue for 45 generations, you'd end up with a big blob of 40 trillion cells, each of which had a genome in it and the ability to make more cells. And so it's obviously not what happens. We're more complicated than just a blob of cells. So somehow, during this process, the cells become different from one another. And so we can phrase this a different way. So just to phrase it more simply, we know we have different kinds of cells. So you have liver cells in your body, you have neurons, your brain cells. And so what makes different cells different from one another? What actually makes these different? And so we know the recipe book, the genome, is the same in every cell. And so what makes it different, so this is a whole topic in itself, so I won't go into it, but you might have heard a term epigenetics, which is about how the genome, this book, gets regulated in different cells to make those cells different from each other. And so a simple analogy here would just be, you could imagine that it's the same recipe book in every cell, but the pages are getting dog-eared or marked up in different ways. And so in one cell, you might have one set of pages that are flagged, or recipes that are active, if you want to put it that way. And in a different cell, you might have a different set of recipes that are active, right? And let's say this is a neuron, and this is a liver cell. And so this you're using the same ingredients to build different things. And so I'm just going to keep going with the meal analogy here. And so you might have the same ingredients. Both of these use bacon, right? But you end up with lunch or breakfast depending on which pages of your recipe book you use, right? And then so some of the ingredients are the same, some of the ingredients are different, right? Okay, so it's the same recipe book, just used in different ways to construct different kinds of cells in your body. Okay, so just to reiterate this point, so each genome encodes a program of cell lineage, right? So part of the program that's in your genome is just how to keep a cell alive, but probably a bigger part of it is how to take that cell and have it divide into something that works as a functional living organism. So a fish or a human. And so it's really the sequence here, the differences between the sequence of a fish and a human that give rise to how they turn out, right? Okay, so how do we study this? How do we study cell lineage? How do we study these patterns of cell division that give rise to different kinds of cells in different kinds of species? And so the simplest way to study something is just to watch it, right, with your eyes. And so people have been interested in this problem of cell lineage and studying it with their eyes for over 100 years. And so in the late 1800s, there were a few scientists, so two of the most prominent ones, August Weismann and Edmund Wilson, are listed down here. And they just watched cells dividing. And so obviously, you've got to work with an organism where you can actually see it. So the development has to ideally happen outside of a mother, for example, and has to be ideally a transparent organism that's see-through. And so, but if you have something that's see-through and you can put it under your microscope and look at it, maybe you can actually watch one cell dividing into 2, into 4. And so these were the first scientists to draw trees of cells, and you can see they have names for each of these cells. Maybe they're labeling these as, well, these ones are in the front, these ones are in the back, these ones are in the side, and so on and so forth. And so the organisms that they're looking here are the sea snail on the left, Triton, and the worm, a kind of worm, Nereus, over here on the right. And so you can only go so far with this. At some point, these stop being see-through, right? And your eye can only do so much. The cells get really small. Microscopes in the late 1800s were only so good. And so they did what they could. And there are big old books that you can still dig out from the library where they're sketching these things out. Okay, so fast forward about 100 years, and kind of the biggest thing in this area that's happened in the last, in the past few decades, has been work by this man, John Sulston, Sir John Sulston. And what he did was he was at the, I believe he was at the MRC research lab in England when he did this work. He took this organism called C. elegans, which is an organism that we still study today quite a bit. And there's a couple things that are unique about this. And this is a kind of worm. If you a rotting fruit will in your backyard might well have some C. elegans chewing through it. So the things that are unique about it, one, is it's pretty small. So the whole adult C. elegans only has about 1,000 cells. Second, it's see-through the whole time. So you can watch it developing from 1 cell to 1,000 cells. So you can watch the entire process. And the third thing is it happens the same way every time, right? So if you were to look at lots of different worms, lots of different C. elegans, they all do the exact same thing to go from 1 cell to 1,000 cells. And so Sulston, for about 2 years, would sit in a dark room with a microscope and just watch this dividing, and then he'd look at another worm and watch it dividing, and so on and so forth. And he did this day after day after day, and he would take notes along the way. And so you can imagine these are actually some of his drawings here on the left. And as he's scribbling down what he sees, he's building up a tree of cell divisions, right? So you go from, we're just showing part of it here, but if you start from this ancestor, you've got a division, and then you look a little further, and you get those cells dividing. You look a little further, you get more cells dividing, and boom, boom, boom. And so on. He builds up the tree just by watching and taking notes, and then actually trying to reconstruct what happened. Okay, so after 2 years, it may have actually taken, I think, longer than that to do the whole thing. He actually got it all. And so here he is looking much older after those 2 years. Actually, I think that's actually a much later picture. But it's not just that he had the tree. He knew who every cell was. He knew which of those cells, which cell was in the pharynx, which cell was in the gut, which cell was a brain cell, and which cell was which brain cell. He knew all of it. And he knew where they were. And he drew this fabulous tree. And so he has this nice quote here, which I think summarizes the accomplishment very accurately. This report, when he reported this in 1993, marks the completion of a project began over 100 years ago, right? The determination of the entire cell lineage of a nematode, or nematode means worm. Okay, so that's pretty cool, right? Okay, so what about other organisms, right? What about large, non-transparent species? So even if we had the patience to sit in a room like he did for who knows how long, we couldn't even do it because we can't actually see this process because things become non-transparent pretty quickly. So it doesn't mean that people didn't try to study this. They really did try, and they started, about 100 years ago trying. And so I'm not going to talk about all the ways that they tried, but I'm just going to try to summarize the main thing that they did. And so what you could do, so you've got this process of a cell dividing, and then eventually some set of cells giving rise to a heart, or lungs, or a stomach. You could imagine taking a very early embryo and somehow marking one of the cells, right? And so there's a lot of ways to do this. The simplest way is to just literally stick a needle into it and inject it with dye. And then the descendants of that cell, the kids of that cell, their grandkids, their great-grandkids, will carry some of that dye. And so you look at the organism. This wouldn't be a human, but let's say it's a mouse or something like that. You look at it much later in development, and you ask where those cells ended up. And then you can infer that all the cells down here are the descendants of that cell that you injected back there, right? Okay. So you're asking where the descendants of this marked cell end up, right? The problem, this is a great approach, and people learned a lot from these kinds of experiments, but there's lots of missing information here, right? So, ideally I would know everything that happened, but I'm injecting something here, and then I'm looking at it here. So everything that happened in between is kind of obscure to me. And so there's some tree that happened between this one cell and all of its descendants, but I don't know anything about who these cells were on the intermediate parts of the tree, where they were, or anything like that, right? So I'm only able to see the end and know the beginning. And so just to show what data like this might look like, so here, this is a mouse embryo, and let's say we've marked, at an early stage, we've injected dye or somehow otherwise marked particular cells. Here we're marking it genetically. But you can see that all the descendants of this one cell here, you can see all of them in this cluster here. You can see the descendants of another cell kind of scattered throughout this region here. And so it's useful information. It tells us something about development, but we don't know the relationships between these cells. We don't know how they're related to each other within that big clump. We just know that they're related, right? We don't know who's sibling and who's cousin and so on and so forth. So the question is, so we have this for worm. How do we get the big tree for other things? How do we get the big tree for a fish? How do we get the big tree for a human? Right? Okay, so now it's very appropriate that my PhD advisor is here in the back. This was supposed to be my PhD project. So I joined George's lab sometime around the end of the last century, and I met with him in 2000 as I was starting my PhD, and he gave me this packet of ideas, right? And I actually, his 60th birthday was a couple years ago, and I turned my office upside down looking for the packet of ideas because it was such a great thing, and I couldn't find it. And then this morning, I just walked in my office, opened the drawer, and there it was, magically. And so the amazing thing about that list, and George, I'll send it to you later, is the ideas are all insane, right? So just as an example, build mirror image life was one of the ideas, right? Which is, George, it's close, we're getting there. But the point is, at the time, as a graduate student coming in, it just seemed like a set of totally crazy ideas. But I think this is one of the remarkable things about George in particular, is that many of these ideas have come to pass in the last 20 years, right? Which was longer than I would hope my PhD to be. But it is kind of amazing to look back at this document and see how many have come to be, including things like next generation sequencing. But one of the ideas that we talked about in that meeting was how you could record information for these kinds of trees. And so, right behind that packet, there was another sheet of paper that I'd apparently printed out, which was my email back to him about how to actually do this, and laying out in a few pages a couple of ideas to do this. And so I hung on to this, I think, as a mark of, I don't know if it's shame or a badge of honor or something like that. But I spent the first 6 months of my PhD trying to get this to work. And then after I presented at lab meeting, a postdoc sat me down and convinced me I should move on to other things, which I did, but I clearly was still upset that I didn't get this to work. And so many years later, a student in my lab, Aaron McKenna, the one who defended last week, kind of came back to this and put a few years into it, more than my 6 months. And that's what I'm going to talk about for the rest of this. Okay, so changing topics entirely. But I think it's also a nice illustration of how long things can take to get to work in science, and how good ideas, even if they seem crazy at a moment, can 10 or 20 years down the line, if they're good ideas, they'll eventually mature and come to fruition. Okay, so how many people have heard of CRISPR? How many people totally understand CRISPR? Daniel, you got it? You're, okay. This is the only one. So I'm going to try and, this could be a whole lecture of its own, so I'm going to go through it pretty quickly, and just kind of give you the high level. So there's three main things you need to know about CRISPR. So one is what it is. So no one invented CRISPR, right? Evolution invented CRISPR. It's the immune system of bacteria. So bacteria have viruses that invade them, called bacteriophages. And they do it by spitting their DNA into the bacteria, and it then kind of takes over, hijacks the cell. And CRISPR is a way that bacteria have evolved to recognize the DNA of bacteriophages and to cut it up, right, to stop the infection. And so the second thing you need to know is it was discovered by scientists at a yogurt company in 2005. So it's quite recent, but I think it illustrates the value of kind of truly random exploration in science, right? These guys were looking for better ways to make yogurt cultures and stumbled upon this. And then a few years later, this system from bacteria was repurposed or borrowed and put into human cells or mouse cells or other kinds of cells. So you could take this system out of bacteria and put it in a different kind of cell, and it still worked. But now for a different purpose. So no longer defending against viruses, but instead as a kind of programmable scissors. And I'll talk a little more about what I mean by programmable scissors in a couple slides. So also notably, since we have George was actually one of the key people in kind of getting this to work in mammalian cells. Okay, so CRISPR, I think more so than anything in my life, at least in my adult life since the Human Genome Project has been in the news. Someone in my lab found this video of someone trying to illustrate how CRISPR works and everything important about it using only food. And so instead of trying to explain it, I thought I'd just show this because it's so good.
You know that it's a revolutionary new tool for editing DNA, but do you truly understand it? Say you're on a date at Red Lobster and your date suddenly asks, how does CRISPR work exactly? Would you be prepared? By the end of this video, you'll be able to answer with a confident yes. Like splitting the atom, this advance in genetics has profound implications for the future of the human race. CRISPR could be used to remove genes that cause the deadliest forms of cancer, or insert genes that resist HIV and other illnesses. But it could also lead to abuses, including eugenics, offspring customization, and the breeding of human beings for nefarious purposes. CRISPR has two components, Cas9, a protein that snips the gene, and a piece of guide RNA, which pinpoints the target gene. The guide RNA directs the Cas9 protein to an exact portion of a DNA sequence, say a gene that causes blindness, and cuts it out or replaces it. But CRISPR can also go off-script, chopping up DNA in unexpected places, creating so-called off-target effects in the patient. So after undergoing gene therapy for blindness, you may also discover that you're now left-handed, lactose intolerant, and more open to polyamorous relationships. Finally, if CRISPR were used on a human embryo, these off-target effects could be passed on to every future generation, possibly creating a new race of freakish telekinetic mutants.
That's how CRISPR works.
Okay, got it? So kind of the key image in there was this one. So, which is this idea that so it's programmable scissors for cutting the genome, right? That's coming back to that concept. So the lobster claw here, that's the Cas9. That's the programmable scissors. So how do the scissors know where to go? So I mentioned that the genome is 3 billion letters long. So somehow these scissors need to know where to cut. If they cut everywhere, you're going to have all these problems. You want to just have them cut in one place. And so you also have this targeting system. This is called a guide. And the combination of the Cas9, the lobster claw, and the guide, the laser pointer, allows you to find the right place and make a cut. Okay? Okay, so now you can take this and you can put the system into, as I mentioned, even though it's borrowed from bacteria, you can put it into a mammalian or a human cell and cut the genome. This is like finding a very specific place in the recipe book and cutting with your scissors. And then what will happen, you can ask, right? So it'll find a place and it will cut. So what happens when the genome is snipped? So evolution has dealt with this problem before. Breaks happen in DNA. And so fortunately, there are recipes in this book that are instructions for how to fix breaks, right? How to deal with a break. And so, okay, so let's say you have your DNA here. So this is just, if you look closely, there's A's, G's, C's, and T's there. This is just a stretch of DNA. And so the Cas9 and the guide RNA find this place where they're supposed to cut because they have a match between the guide and the bases that are here. They look for that match. When they find the match, they cut. And your body is actually, your cells are really good at repairing DNA. They're really good at perfectly repairing it. So usually what happens is it just gets fixed. And it goes back to the exact same sequence that it was before. But then it just sees it again. It sees the same sequence there, so it cuts it again. And then it fixes it. It cuts it again, and it fixes it. So it's this little battle where the cell is repairing it, and the Cas9 and the guide are cutting it. And you keep going until eventually, at some point, the cell makes a mistake in fixing it, right? And so you can imagine a couple different kinds of mistakes. So I'll just show two here. You can imagine that a mistake where, by accident, while it's trying to fix this, it leaves out a couple of the letters, right? And this is called a deletion. So you've lost some letters that are supposed to be there. You can also accidentally insert a few extra letters while you're doing this repair. So you end up with an insertion. Right, so basis is letters. That's the A's, G's, C's, and T's. So these are the kinds of things that can happen. And then as soon as that happens, you no longer have a match. Right? And so your Cas9 and your guide that are looking for where to cut, they can't recognize this anymore because it's not the same sequence anymore. And so they stop cutting. Okay, so it's an irreversible change in the DNA. Make sense? Okay. Okay, so here's the where it kind of comes together. So and just to highlight so Aaron and also Greg Finley were the two graduate students who really made this all happen. So now imagine that the part of the genome that we're targeting is in this first cell, the zygote, right, the cell that starts an organism, and that we literally stick a needle into that first cell, and we inject the Cas9, and we inject the guides that are going to cut at a specific place. And so now, while the organism is developing, we have cutting going on in the genome. Okay? But we've done this in such a way, we're injecting the guides that will target a very specific place. So they're not just cutting all over the place, they're cutting exactly where we want them to cut. And so now you can also imagine that if it's the same, again, it's the same genome in every cell. So this genome is the same as this genome. But when CRISPR is cutting, the repairs that happen, the mistakes that are made, will be different in different cells. Right? So here, you might get a deletion in this cell, and here in this cell, you might get an insertion. So all of a sudden, these genomes aren't the same anymore. Right? One has got a deletion there, one has got an insertion. And because these marks are irreversible, because we've basically made this change and it's going to get passed on, all the descendants of this cell will have this mark, and all the descendants of this cell will have this mark. Right? So different mutations will happen in different cells. Okay, so now if I just had one of my lobster claws, right, here, this would be over. This would be done, right? We've mutated both of these two cells. There's nowhere left to cut, and so you're just you'll grow up, and then you can tell all the cells that came from this ancestor apart from all the cells that came from this ancestor. But now what if we, instead of having one targeting guide, we have 10 of them, right? So now we're making cuts not just in one place, we're making cuts in lots of places, all within the same part of the genome, okay? And so if we do that, we have different changes accumulate in different cells. And so now you can see the tree over here, and you can see different things happening, right? So for example, in this ancestor, you had this mutation, and then that cell divided, and then this ancestor, but not this ancestor, had the second mutation, and then this one right here had a third mutation, right? And so they're accumulating mutations over time, and we can use the patterns of mutations to basically reconstruct a tree about those relationships, right? Based on sharing of mutations. And so this can go on and on, right? We can have other ancestors and other descendants and different cuts in different places, and that's basically how it works. And so on top of that, we can also infer things about ancestors, right? So let's say we inject these reagents, we inject this Cas9 enzyme and the guides at the single cell stage. We let the organism grow up, whatever it is, a fly or a fish or a mouse, and then we look at these at the end, and then we make a tree, right? So we never actually looked at anything in between, but we can still say something about the ancestor. So for example, we can guess that this ancestor had these two mutations because all of its descendants have those two mutations. We can look at a different ancestor and say that it had these three mutations because all of its descendants had those three mutations, right? And then kind of getting more to the point of why we're doing this, we can also learn how different kinds of cells are related. So for example, if we saw that all of these barcodes ended up, I'm going to call these barcodes, all of these ended up in the cells that have these barcodes ended up in the lung, and the cells that had these barcodes ended up in the stomach and eye and brain, we could say something about the relationships of these organs to one another, right? So here we would say that the eye is more closely related to the brain, and the lung is more closely related to the stomach, right? Okay, hopefully folks are still with me here. So now how do we put this together? How do we actually do an experiment that's interesting and useful? And so the place that we tried to, once we had all the pieces of this working, the place we tried to actually do something with it was the zebrafish, right? So zebrafish is a kind of fish. It's a model organism in genetics, meaning that it's one of the main organisms that we study as biologists. And one of the things that is appealing about it is it's a vertebrate, right? So like humans and mice, it has a vertebrae, and so it's closer to humans than, let's say, a fly or a worm, and therefore maybe more relevant for understanding human disease. Okay, so to do this, we teamed up with a fish lab. This is Alex Shear at Harvard, and his postdoc, Jamie Gagnon, worked with Greg and Aaron on actually getting this to work. Okay, so we have two fish, right? They mate. We have a single cell that we could then inject with Cas9 or other things, and then we let that single cell develop. It goes through some of these early stages very quickly, and I'll show some videos of this in a few slides. It goes through these early stages very quickly, already looks kind of like a fish after a day or two, and then 3 months later you get an actual adult fish, right? And one of the nice things about this, in terms of the experiment that we wanted to do, is that there actually is a big fat egg that we can inject things into, right? So Jamie is very experienced at this and was able to go, under a microscope, stick a needle into that egg and inject the Cas9 and the other things that need to be there to start cutting the genome. Okay, so I'm now going to show a couple just videos of fish development, so you can watch this because it's pretty amazing. So there's a great quote here. So of this process called gastrulation. So the quote is, it's a well-known quote in developmental biology. It's not birth, it's not marriage, it's not death, it's gastrulation, which is the most important time in your life. And so gastrulation is when you go from kind of just a sheet of cells that are basically all the same as each other into a bunch of what are called germ layers. I'll play this while I'm talking. So here we're going from 1 cell to 2 cells to 4 cells to 8 cells, 16, 32, 64, kind of lose track of it there. But somewhere in this process, gastrulation will happen. It'll be a little easier to see on the next one. So the cool part about this video is that they were able to mark the genomes in every cell. So the yellow dots that you see lighting up are the genomes in each cell that are kind of like labeled with a marker. And so you can see it going on. So I'm going to stop this one because it's not as cool as the next one. But you get a sense of what this looks like. So this was probably a couple hours of development. So this is going to be now the first 24 hours of development condensed into about a minute. This is all one day, right? And you can't see the cells as well here because of the kind of microscopy, but these are 4 different zebrafish developing. And what we saw on that last slide is developing on the surface here. So you can kind of see it there, developing. And at some point, you have this process of gastrulation happen where the different germ layers form. So these are names like ectoderm, endoderm, and mesoderm that will then go on to give rise to the ectoderm will give rise to your brain and your skin. The endoderm will give rise to your gut. And they're swimming, right? 24 hours later, they're zooming around there, right? It's amazing. And then the mesoderm will give rise to your heart and other your muscle and so on and so forth. So gastrulation is important. The reason he said this is because so many things can go wrong in that early process, right? And you all made it, so congratulations. Okay, so how do we do the experiment we're going to do? So we engineered this barcode into the genome of the mother fish. Okay, so we got mom over here, and in her book of life, we've stuck in an extra page. That extra page is basically a bunch of addresses for our lobster claws. Right? So they're going to go just to that page and make their cuts. Right? And then we mate this fish to another fish. We get an egg, fertilized egg or zygote, and then we inject into that egg the Cas9 and the guides. And so these guys are going to go, and they're going to, through all this giant book of life, they're going to find just this place, and they're going to start making their cuts. All right? And so we do that, and then we just wait. We step back and we wait. And the fish develops. So it goes through these various stages, and eventually an adult. So we have to wait 3 months, so we're a little bit impatient. So we took some of the earlier embryos and tried to look at them early on while we were waiting for some of the adults to grow up. Right? Okay, so the next slide I'll show an example where we harvested here. And you can't really, we can't dissect the brain and the neuron. We can't dissect different parts of the fish at this early stage. So we're just taking the whole fish and looking at it kind of all together. Okay? And this is the kind of stuff that we see when we look at that. So what I'm showing on the next slide is going to be this barcode and kind of seeing what happened to it in different cells. Okay, so it's a bit of a complicated slide. I'll just spend a minute walking through it here. So every line here represents a different thing that we saw happen to the barcode. Where the red bars are deletions, so parts that got deleted out, and the blue bars are insertions, things that got stuck in. And so what's shown over here is how often we're seeing a given barcode. So the ones up here we saw the most, and the ones down here we maybe only saw in one cell. And this is just showing how often we're seeing cuts at different places along the barcode. And so one of the cool things you can see here is that about almost exactly half the cells have an insertion right there. And it's all the same insertion. Right? And so probably what happened is that when this divided into two cells, this insertion happened in one of those cells, and then all of the descendants of that cell in the fish, or the fish that we harvested, have that mark, right? And then everyone who doesn't have it is probably the descendants of that other cell, the second cell, right? And so from these kinds of patterns, and comparing the patterns to each other in different cells, we can build trees, right? So this is just a part of a tree, a bigger tree. And so here we can see that the reason that we put all of these guys together, all of these cells together, is that they all share these same edits. And then we have different parts of the tree where we put them together because they all share a subset of the edits, right? So every cell here has this big deletion in the middle, but this group up here is unique in that it also has this other deletion, whereas this group has this insertion, and these other groups have different other mutations. And so we're building up or inferring a tree based on what we see at the end. And the fun thing is we can make really big trees with this method, right? So this tree is big enough that we couldn't fit it all up and down, and so you go down to the bottom, and then it starts over, and then it starts over here, right? So it's a very wide diagram here. And so in total, we see about 1,000 different kinds of barcodes, right? We call these alleles. And we might be looking at more than 1,000 cells or more than 1,300 cells, but sometimes we see the same barcode over and over again, right? And part of the reason is that we injected the Cas9 and the guides in a single cell stage, and the cell divided, and eventually there just wasn't enough of it left, and it probably stopped cutting, right? And so we've marked the cells in those early divisions, and then they just kind of keep dividing into it as far as they get, okay? And so in addition to being able to build these big trees, we can do this on a lot of different fish. And so here we're so every one of these is an independent tree or cell lineage that we've made. They're really hard to see, but we've made them from different fish at different stages of development, right? So these are early, very early, and these are now after 3 days, and we can build these trees. And so the downside of this, or the thing that makes this not terribly useful in this form, is that we're building these great trees, but we actually don't know who any of these cells are, right? So we might see these cells, but we don't know who's a brain cell and who's a liver cell. We just have a bunch of cells from an embryo that we mashed up, right? And so to look in more detail, we had to wait, right? And then finally, after 3 months, we had an adult fish that was big enough that we could dissect it. And so now we can take different parts of that adult fish. We can take the brain, we can take the right eye, we can take the left eye, we can take the blood, the heart, and we can look at these barcodes in different organs and compare them to each other, right? Okay, so I'm going to just go straight to the big tree that we made from the adult, which is here. And it's like the tree I showed you before. It's another big tree. But now we have these bars on the outside, which are basically showing the different organs that we see different barcodes in, right? So, and it's a very non-random pattern. There's some barcodes that we see only in some organs, other barcodes that we see only in other organs. And that's interesting, because we can look at what those patterns are. And so this is the big lineage. Every part of this tree we'll call a sublineage. And so here I'm just looking at part of the tree where this one ancestor gave rise to all of these cells. And you can see we know that because they all share these mutations. And then they have other mutations. But we can look at the organs in which we see these barcodes. And so some of them, if we look at the whole set, they're in every organ. But now if we just look at part of it, if we just look at this top part, they really tend to be in the heart and the skin, right? If we look at just these two barcodes, or this one set of barcodes here, they're only in the eyes, and they're half in the right eye and half in the left eye, right? So probably with some ancestor that contributed about equally to each of the eyes. So the other really interesting thing that stood out to us from this data was that if we looked at each organ, it was dominated by the descendants, the children, of just a handful of ancestor cells, right? So this is looking at blood, and this is the abundance on the right, and this is on a log scale. So basically what this is showing is that there are just 5 cells, 5 ancestors that were the ancestors of almost all of the blood in this adult fish, right? And so somehow, these 5 cells very early on, of the thousands of cells that were present in this early embryo, gave rise to all of the blood that we're seeing in the adult fish. And we don't just see this in the blood, we see it in other organs too. So if we look at the brain, we have about maybe 20 ancestors or so that dominate the adult brain, right? And it's a different set of ancestors than the ones that gave rise to the blood. And so this is just a way of showing that information. So this is a cumulative plot where we're looking at the most frequent allele, the second most frequent, the third most frequent, and then we're saying how much of the organ do they represent. And so this is blood. Basically what we're showing is if we just look at the first 5 or 6 alleles, about 100% of blood is explained by those 5 or 6. So now if you look at the brain, is it the brain? No, that's the gut. It's similar, right? Where you also have a relatively small number of cells giving rise to the gut. You can look at, this is the overall adult, where still it's a surprisingly small proportion of the ancestral cells that give rise to the adult. Actually, I think that's the brain. That should be the brain. And we can do this for every organ, and we see the same pattern, where very few cells are giving rise to each organ. Or we can look at the adult altogether. And so this is kind of surprising because it suggests that somehow, whether it's early in development or whether it's late in development, the descendants of a few ancestors are kind of taking over each organ. Now, if we look earlier in development, if we look at those embryos, they don't show the same pattern, right? We see kind of a diversity of different cells contributing about equally. And so somehow you go from this pattern in the embryo where you have lots of ancestors about evenly represented to this pattern in the adult where you have bigger clades or bigger kind of domination of different organs by different sets of descendants. So kind of a different way of showing this graphically is to borrow from an artist, Mondrian. So kind of we call these Mondrian plots so here we're showing blood where the descendants of just 5 ancestors explain almost all of blood. The brain is more complicated than blood, but still you have a relatively few number of ancestors that are dominating. And then we can look at other things too. But then if you look at the embryos, they're much more even representation. Okay, so this is kind of a proof of concept. We've done this big lineage, but it's certainly well short of a complete fish. And there's all kinds of interesting questions that one can ask as we try to build bigger and bigger trees. And so one of them, I mentioned early on that the worm, it's always made the same way, right? It's always the same lineage. And so we know in humans and mice and fish that it's not always the same. You can have a genetically identical fish or human, and somehow you have this different pattern of cell lineage give rise to something that looks pretty much the same. So take like twins, for example. And so how many ways are there to build a zebrafish, right? And so we just actually got, going to acknowledge our past and future funders. So we just got a very generous grant from the Paul Allen Frontiers Group to continue this work into the next 4 to 8 years. And the idea here is to do something similar to what we just did, but to do it much more comprehensively for an entire fish, and to also do it for the mouse, right? With the idea that these trees will really hopefully unlock a lot of knowledge about or understanding about how vertebrates develop, right? Okay, I'll just close on a note that it's kind of a fun thing to notice that life is full of trees, right? So you have trees of species, you have family trees, and you have trees of cells, and we kind of all of these trees are important for kind of giving rise to the diversity that is around us on this planet, right? So I'll stop there and just thank these folks. I think I already talked about all of these guys. So Marshall Horwitz is another investigator here at UW who's been a longtime collaborator of ours in this area. And this is a figure that my daughter made of a scientist doing this experiment for me last year when we were kind of in the throes of it. Okay, thanks, and happy to take questions or look forward to chatting afterwards.